Okay, so starting Unit 3, we are going to kind of go into energetics, and to begin that, we need to learn about enzymes. So enzymes, essentially, are proteins, so it's one of the specialized functions of proteins that are made. It can also be a combination of RNA too, but their job is to help or facilitate chemical reactions. So by that regard, they are biological catalysts. They speed up the rate of a reaction without themselves getting used up. So they can kind of run around making reactions go faster without being depleted in any way. That's why under the second bullet point, it says there's no change in free energy. What it essentially does, and we'll learn more about this term later, is that it kind of reduces the activation energy. It lowers the amount of energy that you need to put into this equation or reaction to actually happen. So by doing that and lowering the amount of energy that it needs to take to happen, it makes it go faster. And if these protein enzymes were not around, then we wouldn't really be able to do most of the biological reactions in the speed with which we need to do them to stay alive. They are highly specific, so it's kind of a very unique situation, and we'll learn about lock and key and induced fit, but most of the time we think of it as like a lock and a key. There's one enzyme to one substrate. So therefore, our cells have thousands and thousands of enzymes to make this magic happen and keep us alive. Some of the vocabulary that you're going to need to know: a substrate. So that's the thing that the enzyme is acting on. When those two things come together, it is called an enzyme substrate complex. And really, it's a temporary association because the enzyme is floating around, or the substrate's floating around, and then they magically find each other, they bind, and then the enzyme helps the reaction take place. So they're stuck together for the temporary moment of making that reaction happen, and then the enzyme will go off and find another substrate to do its thing. The product is the end of the reaction, so what is the end result of the substrate? And then the active site is really where those two things come into contact. The exact spot where the enzyme and the substrate connect is called that active site. So here's just kind of a picture of what that looks like. In this case, the end goal would be sucrose turning into or being broken down into glucose and fructose. So sucrose is a disaccharide, and we used that in our diffusion lab, and it is going to kind of be floating around, and the enzyme that's highly specific to that is going to float around. Then in number 2, they stick together to form an enzyme substrate complex. It will bind to the active site of the enzyme, and then in this case, hydrolysis is going to happen because you see the water entering, and it's going to split up those two things, and they're going to pop away as glucose and fructose as products. And now the enzyme is just as it was when it came in and is going to go off and do the same thing to another sucrose molecule. In terms of properties of enzymes, as I mentioned, they're reaction specific. So each enzyme that we have is specific to a very singular substrate. It's not consumed in the reaction, so it doesn't get depleted. A single enzyme molecule can catalyze more than thousands and thousands per second. That's kind of crazy, but we need that in order to stay alive. And they are affected by cellular conditions. So there's lots of things that can happen to the surrounding environment. In this case, I'll just say like our body in general or the cell. But those changes in conditions can affect a protein of any kind. We kind of learned about denaturing. The same is true for an enzyme because it is a protein. So if you go wildly outside of the normal temperature range or the pH range or salinity, which is salt, you then affect the ability of the enzyme to do its job because most likely you're going to affect its structure. In terms of naming an enzyme, they always end in ase, and they are usually named after whatever their substrate is or whatever reaction that they're making happen. So for example, sucrase, its job is to break down sucrose. A protease is a very general term. There's thousands of them, but it's to break down proteins. Lipases break down lipids. We've also learned in the past DNA polymerase. Its job is to build DNA or make a polymer out of DNA, so it's the thing that adds the nucleotides. We also have pepsin, which is an enzyme that lives in our stomachs, and its job is to break down proteins or peptides, so that's why it's called pepsin. That's one unique instance where it doesn't end in ase, but by and large, if you see something that ends in ase, it's an enzyme. So there's kind of two ways that this magic happens. The most traditional sense is lock and key. It's kind of a simplistic model, but the idea is the substrate is the exact shape that the enzyme has kind of a hole for, and that's considered the lock. And then they just magically fit together, and hydrogen bonds form between them, and that's what makes the enzyme substrate complex. A more realistic and accurate model, though, is not a specific lock and key. It's more of what we call an induced fit. So the enzyme itself is kind of roughly shaped where it needs to be, and the substrate, when it comes together and actually binds to the enzyme, there's kind of a conformational change that happens. The enzyme and the substrate change a little bit, and then they bond together, and it increases the binding when they come together. How do enzymes work? Well, as I mentioned previously, what they do is they lower the activation energy. So the amount of energy to kind of activate the reaction or make the reaction take place. And so that in that way, you don't have to spend as much time gathering energy to make it happen. So by lowering the activation energy, you can make it happen faster. So that's really why they're speeding up reactions. And enzymes don't only break things down, they are responsible for synthesizing some things. So the active site kind of orients substrates in the correct position, and the enzyme can also bring substrate closer together and synthesize certain molecules. Enzymes also work in digestion. So in this case, similar to sucrose being broken down into glucose and fructose, the active site kind of binds and then puts stress on those bonds. In this case, there was hydrolysis, but essentially the enzyme is putting stress enough to break the bonds between molecules. So it can either bring things closer together and help them bind, or it can put stress on them and make them break. Overall things that can affect the ability of an enzyme to function. As I mentioned previously, things like temperature, pH, and salinity are some of the easier ones. But other things, enzyme concentration, so the amount of enzyme that happens to be floating around, substrate concentration, the amount of substrate that the enzyme can act on, as well as other things that might be floating around, activators and inhibitors that can facilitate or hinder the enzyme from being able to do its job. So I'm going to kind of talk about each one of those more specifically. Enzyme concentration. And I apologize, the screencast kind of cuts out the very bottom of the slides, but in this case, the x-axis on the graph there is enzyme concentration. So as enzyme concentration increases, you can kind of see here, across the x-axis, the reaction rate increases. This does level off at a certain point. So the idea is the more enzyme there is, well, then the more likely that that enzyme is, so that's what these red guys are, the more likely that it's going to be present near a substrate. So therefore, it can make those reactions happen faster. But then at that point, there's a certain leveling off, because no matter how many enzymes you have, maybe then there's just not enough substrate for it to continue to do its thing. So it kind of reaches a maximum possible rate of reaction. The same is pretty much true for substrate concentration. As the substrate increases, well, then that means that the substrate is going to hit into the enzymes a little more often. And then in that regard, the limiting factor here is the amount of enzyme that's available. So it's going to speed up the reaction up to a point, and then it's going to kind of be saturated. You can continue to add more substrate, but the enzymes are already working to their maximum capability, so you can't make things go really any faster. Temperature. just like, we as people kind of have an optimum temperature, enzymes have optimum temperatures too, where basically they're just the most efficient. They can do the greatest amount of collisions, reactions, and enzymes. So for us in our body, most of them really like to work around body temperature, somewhere between 35 and 40 degrees Celsius. If we get too hot, then that increased energy can start to denature proteins in general, and in that case, enzymes. That's why having anything, a fever over like 104 to 105 is so dangerous, because that can happen. Too cold is also a thing. And in this case, the molecules are moving slower. So you are slowing down the ability of the enzyme to come in contact with the substrate. It's not to say that it won't work at all. It's just not as effective. So here's kind of what that looks like. In this case, we're showing you a human enzyme, so it's happy at around 37 degrees Celsius, and if you get hotter than that or colder than that, then it starts to lose effectiveness. Other types of bacterial enzymes, for example, the ones that live in the crazy archaea that are in hot springs, well, it makes sense that they have optimum working temperatures at those hot spring temperatures. And if you were to cool it down to kind of our own body temp, which is weird to think about, but if it's that cool, then those enzymes don't function properly. Same thing if you were to magically go hotter than that as well. pH has a very similar curve and explanation as temperature. Enzymes have an optimum pH. In this case, most of ours in our body work between 6 and 8. There are exceptions, like pepsin is a stomach enzyme, so our stomach pH is roughly between 2 and 3 to be able to break things down, so it makes sense that it wants to be happy at that 2 to 3 pH range, whereas our small intestines has things like trypsin, where the natural pH is around 8. And you are able to change pH by adding or removing H+ protons. The more of them you add, the more acidic it becomes, and the lower the pH is. If you remove H+, then the higher the pH becomes, and therefore more acidic. But by doing this, you're kind of hindering the ability to make hydrogen bonds. So therefore, you are essentially disrupting the shape of the enzyme, and we know structure affects function. So if you're changing the structure by removing H's, then you're inherently then hurting the function. Next, we have salinity, salt concentration. Similar format, changing salinity adds or removes cations and anions, so positively charged things and negatively charged things. Just like pH, pretty much exactly like pH, you're changing the structure. By changing charged things around and moving it around, you are changing the structure of the enzyme and therefore potentially changing the function. Enzymes do not like extreme salt. So the Dead Sea, for example, is called the Dead Sea for a very specific reason. Even enzymes don't like to work there. Things that can regulate enzymes. So whether it's speeding enzymes up or slowing enzymes down. The first in this case are helping. So these are activators. They enhance the activity of the enzyme. There's two kind of ways to do that. One is called a cofactor. So these are non-protein things. They're small, they're inorganic, and they can be ions. I've listed a couple there for you. So we have magnesium, potassium, calcium, zinc, iron, copper. But they are bound within the enzyme molecule. So one of the key examples here at the right is iron, as it is bound in hemoglobin, the oxygen-containing protein, and that can help facilitate some reactions. Coenzymes are another thing. They are also non-proteins, but these are organic molecules. They temporarily or permanently bind to an enzyme near its active site. So it's called a coenzyme because it's kind of a helper. It helps make the magic happen and works with the enzyme together. So a lot of these are actually vitamins. And again, they're organic compared to cofactors, which are inorganic. So NAD, which we'll learn more about later, is vitamin B3. FAD is vitamin B2. And then there's also coenzyme A. All of those are going to come into play when we talk about cellular respiration in a bit. But regardless, they bind near the active site of the enzyme and they can help facilitate reactions. On the flip side, we have inhibitors. And there's many different kinds, but overall, they reduce the activity or hinder its ability to function. So there are competitive inhibitors, non-competitive inhibitors, and then there's irreversible inhibition, which is exactly how it sounds, and then feedback inhibition. So I'm going to kind of talk briefly about each one. Competitive inhibitors, like the name implies, literally compete for the active site. So they are shaped in such a way that they can mimic the shape of the substrate. So the enzyme thinks that the inhibitor is a substrate and binds to it. So when the inhibitor binds instead, it prevents the enzyme from acting on its substrate. Penicillin is an example of this. So it is a competitive inhibitor for the enzyme that helps bacteria make cell walls. And if bacteria can't make cell walls, they die, which is why penicillin is awesome. Disulfiram is another one. It's an enzyme that breaks down alcohol. So this is often used as kind of a detox medication for those who have trouble with substance abuse. And it basically makes them sick because you can't break down alcohol. And so it acts as a competitive inhibitor there. How do you overcome this? Well, in theory, if you just add more substrate, so you just increase the percentage of substrate, that kind of minimizes the ability of the competitive inhibitor to work. So that eventually you have so much actual substrate in there, it can outcompete the inhibitor. A non-competitive inhibitor is similar, except it doesn't actually bind in the active site. It binds somewhere else. And when it binds somewhere else, it causes the enzyme to change shape. And when it changes shape, it can't do its job. So these are often called allosteric inhibitors. They bind to what's called an allosteric site, something that is not the active site. And it still does the same job as an inhibitor, but in this case, it's doing it by changing the enzyme's shape so it can't work. So some anti-cancer drugs are this. Cyanide poisoning is an example of this. Irreversible inhibition is exactly how it sounds. The inhibitor itself permanently binds to the enzyme. It locks itself into the active site and doesn't ever come out. So basically it prevents that enzyme from ever being able to work again. This can be allosteric as well. If one binds permanently to the allosteric site, it will cause a permanent shape in the enzyme, and then it can't work anymore. This is a huge deal because if it's a necessary enzyme, then it can pretty much cause death. So examples include nerve gas, sarin, and many insecticides. They irreversibly bind to a particular enzyme, and then that enzyme can no longer function. Here's just an example of what conformational changes might look like and what an allosteric regulation looks like. So here, for example, you might have an active site that's in this little crevice here. And if there's an activator, it might block in on this allosteric site and lock it into open position. Whereas an inhibitor might lock itself in an allosteric location and keep it in its inactive form. So when those things bind, the shape may force it to stay active or stay inactive. Okay, so a lot of times chemical reactions of life are organized in pathways. So there's a lot of tiny steps involved to get from one major thing to another. When we start to learn about cellular respiration, there are a lot of steps. So as you can see kind of across the top, there might be 6 enzymes in getting from A to G, or there might be a lot of enzymes in the membrane that go from having our initial substrate to whatever final product it may be. And it kind of allows for intermediate branching points. Instead of having one thing be in charge of everything, it's more like an assembly line. This increased control at each level helps regulate a little bit. again, it's not just shutting down or turning on one thing, but the cell, by controlling different ones at different levels, can kind of control the whole thing. Feedback inhibition is an example where the amount of products that you make can actually turn the system off. It's a way to prevent over making something. So oftentimes it's a negative feedback loop or feedback inhibition. You make enough of the product, and as you get enough of the product, you shut down the assembly line until you don't have enough of it anymore. So one example is threonine, which is the initial piece, can change into isoleucine. So there's a few different enzymes and a few different intermediates, but the end product is isoleucine. And as isoleucine kind of reaches a tipping point in levels and gets high, it actually binds to the allosteric site on enzyme 1. And in this case, it's acting as an inhibitor. So this line here means that it's inhibiting. So acting as an allosteric or non-competitive inhibitor, it can then prevent any more of isoleucine from being made. And then when levels drop to a low amount, they won't inhibit the enzyme anymore, or very infrequently, and you'll make more isoleucine. So this is just a way to kind of overall control the population of what you're trying to make in the cell. And with that, we've completed enzymes.